Hello, my name is Josephine Kelly.
And I'm Noah Seagraves.
This is our final PowerPoint, and it's on the impact of plant-based diets on the gut microbiota. So, as a little introduction, the community of microorganisms, which is known as the human gut microbiota, is essential for immune system function, disease prevention, and digestion. Recent research has shown that diet has a major impact on this activity and the composition of gut microbiota. And there have been a lot of recent research studies done that plant-based diets, such as vegan and vegetarian, are rich in fiber, polyphenols, and other compounds, and that they can positively affect this gut health. And these diets have also been associated with an increased microbial diversity and also beneficial microbial populations. So, in the original experiment that we found, the purpose of the study was to investigate the relationship between long term and short term plant based dietary patterns and its gut microbiome in a big group of Chinese adults. So, specifically, the study aimed to examine how habitual plant based diets and short term plant based dietary intake was associated with the composition of the gut microbiome, and they used stool samples collected from the different participants in 2015. The study also considered various demographic lifestyles and medical factors to better understand these associations. Our reason for looking into this and wanting to kind of explore it more was to address the gaps that were in the current understanding by investigating the impact of different plant-based diets, whether it was high fiber or low fiber, on the gut microbiome. For background research, we found a few different studies aside from our main original experiment, and one was a systematic review of international studies finding that plant-based diets increase gut microbial diversity, particularly promoting growth of different bacterias, which are linked to a healthier metabolic profile. In that other study, they demonstrated that vegan and vegetarian diets specifically enhance the production of short chain fatty acids, which have a lot of anti inflammatory properties. These findings suggest that plant based diets positively influence the gut microbiome composition, which may explain their health benefits. So, for methods on our proposed study, as the independent variables, there were going to be two different plant-based diets, high fiber and low fiber, versus a standard diet, which would be someone's everyday diet with eating meat and vegetables. Dependent variables, looking at gut microbiota composition, inflammation markers, and metabolic syndrome parameters. Control group being the standard diet group. For replicates, we chose 5,000 American adults, with 50% being men and 50% women, ages 35 to 65. Our null hypothesis states that there would be no significant difference in the gut microbiota composition, inflammation markers, or metabolic parameters between individuals following a plant-based diet and those following a standard diet over the 12-week intervention period. And for our main hypothesis, individuals following a plant-based diet would show significant improvement in the gut microbiota composition, a reduction in inflammation markers, and a better metabolic health outcome compared to those following the standard diet over the 12-week intervention period. For our experimental design, we have a randomized control trial with 3 groups. So the first one being low fiber plant-based diet group, which the participants would follow low fiber plant-based diet for 12 weeks. The next being a high fiber plant-based diet group, same thing, but with high fiber over the 12 weeks. And then the standard diet group being the control group, where participants continue their usual mixed diet for 12 weeks.
For data collection, the testing would be done at 2 key time points. The baseline or week 0, where the gut microbiota would be tested using stool samples and they would be collected for rRNA sequencing Our inflammation markers would be blood samples for CRP, IL-6, and TFN or TNF alpha. The metabolic parameters would be the blood tests and measurements of the waist circumference, blood pressure, blood glucose, and cholesterol levels. For the end of intervention test, that would be week 12 or the last week. The gut microbiota, inflammation markers, and metabolic parameters would all be tested the same as the baseline week. This allows for comparisons of the baseline and post intervention data to evaluate the effects of the plant-based diet. For potential results, figure A, which is the top left in the image, the association of the long-term and short-term PDI with firmicutes. And for figure B, that's the top right, two bar graphs, the number of significant taxonomic associations observed in the genus and the ASV level with the long-term and short-term plant-based dietary patterns. The short-term's in green and the long-term is in that orange color. Figure C is the chart at the bottom, and that shows the associations of the long-term and short-term plant-based dietary patterns and its constituents food groups with the gut microbiota. And here's our p-value there. It was 0.001. For potential results, our hypothesis that the plant-based diet will lead to changes in the gut microbiota and improvements in inflammation and metabolic health is very likely to be supported by the findings of the original study, particularly with respect to the long-term effects of plant-based diets. In the event of a lack of support for our hypothesis, it may be due to individual differences, nutrition imbalances, the short study period, and confounding variables like lifestyle, physical activity amounts. Those sorts of things obscure the potential benefits of a plant-based diet for inflammation, gut microbiota, and metabolic health. The significance of these results for our p-value for the microbiota composition and the inflammation markers in the original study suggests that similar findings could be expected in our experiment. For implications, if our hypothesis is supported, it would significantly advance our understanding of how plant-based diets influence gut microbiota and metabolic health. More research would fill important gaps in current knowledge, including the long-term effects of plant-based diets on the microbiome, the specific microbial taxa involved, and the mechanisms by which dietary changes influence inflammation and metabolism. This is crucial for developing personalized dietary guidelines and improving strategies for managing diet-related chronic illnesses like type 2 diabetes and cardiovascular disease. Ultimately, our findings could have significant real-world applications in clinical nutrition and public health policies, promoting healthier diet practices globally. Another implication that I'm personally interested in would be a short-term group, 3 to 6 weeks particularly. I think that would be interesting to see how an even shorter time frame could change somebody's gut microbiota for the better. And here's our resources.